the ligament bioreactor. We're going to talk about actually building it, and we're going to talk about the analysis that comes afterwards, which we started talking about last time. And last time the ending point was that the PCR is a better way to tell what cells are actually making versus what's already in the matrix using staining that we normally would have done. So first I'm going to talk about the bioreactor itself and some of the small changes from last year to this year. All of the instructions are pretty clearly laid out, I think, in the lab manual, but we'll kind of walk through what it looks like today, and I have one sitting here so you can look at it. This one's always going to be available in the lab for reference. So this year the chassis or the actual dish is different than it was last year. Last year we used this kind of large square staining dish, which is over here. This year we're using something smaller. It's similar to this. Couldn't reach the box because I'm short, so brought this one in as an example. So it looks very similar to this. So it's pretty small, compact, but it holds a lot of media still, which is what we said we needed to keep them happy. It is a motor-driven cyclic strain, so we have an Arduino controller that we're going to program to run a servo motor, and the servo goes back and forth and stretches the ligament over and over and over at some sort of periodicity that you program in there. And then the ligament, you guys just started decellularizing the ligament, so that's a pretty long process, lots of rinses and soap solutions to get all the cellular debris out. But once you're done with that, we'll check that it's decellularized fully and then reseed it with new cells, with mouse cells, to start the process. So basically, each group has to build one bioreactor and decellularize one ligament. And then before you actually run in your bioreactors, we'll test them. We'll test them to make sure that they're holding liquid, of course, test to make sure that your motor is working properly, and confirming the decellularization. So what do you think we might do to confirm the decellularization?
Other than implant them, see
To confirm the decellularization? So we took all the cells out of it. How are we going to check that? DAPI nucleus, right? If there's no DAPI, DAPI stains DNA within the nucleus. So if there's no nucleus, there's no cell. If there's no DNA, then we've removed everything, every trace of the cell from the ligament. So we're going to stain with DAPI and look at them before we move on.
So that should show up even if there's DNA floating around, show up on free DNA?
Yes and no. Free DNA, it will stain, but you won't be able to see.
It's not as concentrated?
Yeah, it's just too small. But if there's clumps of it, you'll see it light up.
So is there a way for us to verify that we got all the RNA and DNA?
We're going to say if we don't see fluorescence that we did a good job. It's at least pretty much destroyed. Free-floating DNA isn't going to have a huge issue because it's free-floating, so it'll float away with media and washing and things like that. As long as the nucleus is completely disrupted, you don't see nucleus, there's no cell, there's no nucleus, you're pretty good. So that's what we'll assume. So we'll do a DAPI to confirm decellularization. So you guys might remember DAPI is a super quick stain, takes half an hour. You can just throw it on there, let it soak half an hour, look under the microscope, 10x, 20x, whatever, make sure there's no nuclei there, and then you're good to go. So the bioreactor plans are in your notebook, but I have one right here that I'm actually going to turn on and you guys can come up and look at it if you want to. So it's running right now, so you guys feel free to come up and look at it while I'm talking. So this is when it was built last spring. It's an improvement upon our old design. And like I said, it has this little servo here that is pulling back and forth a programmed amount to stretch this, and it's supposed to be a 10% strain is what we're going for. So depending on the length of your ligament, you may want to adjust how much the servo arm moves so that you get the appropriate strain that you want. We decided on this as our cyclic periodicity, but I wouldn't go much faster than, say, a hertz or so because you don't want to go crazy. But we have ours run for about an hour a day every day. And that's probably enough. You don't want it running all day long. It's a little too much stress on the cells. So that was the determined protocol that we suggest you use. Again, you're always welcome to experiment if you want to change some of these parameters. That's your prerogative and you're welcome to do so, but if you do, please base it on literature of some sort and back it up. But this is just a simple Arduino. This one's run off USB. They also run off DC power supply. Plug them in. We have power supplies, of course. You can plug them in the wall. We have automatic, what do you call it, Christmas light timer that you can program. So it runs an hour a day every day. So that will save you guys some time coming in. But you'll still have to come in and check them to feed them. So this is very similar. Again, it'll just be in a smaller box for less wasted media. This coming here. And right now there's just a piece of tape between the two to kind of show that something goes there.
So, ligaments gonna be longer than that though?
No, about that length.
Okay, because we were cutting them last week, and Haley sent us an email saying we cut them too short. We were kind of trying to tailor to that.
Yeah, your new ones are about that length too.
Okay, so we just cut too short.
You were using really old ligaments that I didn't intend for you to use. I was getting you fresh ones. And I had notified Haley that the fresh ones were coming, but there was some miscommunication apparently. So your new ones came on Friday and she started them for you over the weekend. The new ones are dog ligaments and they're fresh.
So it wasn't that it was too short, it's just the wrong ones.
They're the wrong ones.
They were told they were too short. Oh.
I haven't seen them. I don't know if they were too short or not. I just don't imagine that you could have gotten 4 good ones out of that one ligament. So now we have new ones, fresh ones, dog ones. Yeah. All right, if you guys are done looking at this. So as you might see by looking at this, it's not too complicated. You have to drill a hole here or there, glue some things down, seal with acrylic. So that's what you're basically going to be doing. Last year, Chris did build this little kind of stage that the servo's sitting on just to hold it in place. That's a good idea if you want to add that on to your project, but there's lots of scrap acrylic downstairs to do so, but you don't have to. Whatever you do to kind of house your servo motor is going to be fine. The servo's pretty airtight, pretty moisture tight. It should be fine running in the incubator, but anything you can do to protect it is going to be great. So just kind of consider building it a little housing or a little shelf to sit on. So you'll have access to the fabrication lab for the project, and you'll definitely need to go in there to drill your holes, and so we'll talk about that. But you might need some of the other tools if you're going to be building a little housing. You might need to use the saw or the grinder, and everything down there is use whatever you need to get your project done. So there is a parts list in the manual. I'm still working on getting a few of the parts, but the dishes are here and ready. Like I said, I just can't reach them. So they're here. So before you get started, you'll want to make sure that you do have all the parts that you need. If you break something, if you mess up, I have extras of the dishes, but everything else you'll want to get on your own. None of the parts themselves cost more than a few dollars at most. Probably the hardest to find thing is the little bicycle boot that we have insulating the servo motor connection, but you can buy that at any bike shop. So pretty simple products. We have lots of Arduinos, we have lots of servos, so those are already here and ready for you if you want to start playing with those. Everything is just going to be pretty simple and according to the plan that's in the notebook. So I'm going to put a key up in 3116 in a drawer that you can use to go down to the Fab Lab. The rule with the Fab Lab is you have to have a buddy with you, somebody who knows how to use a phone, because there's not a phone down there. We switched to Link, there's no phones anymore, only phones through computer. So somebody down there has to have a cell phone, know how to use a cell phone, I should say. And it can be a boyfriend, girlfriend, whoever, somebody down there who's going to be listening for you to make sure you're okay and can call if you need help. That's basically the only rule. There are instruction manuals down there on how to use all the tools. I can briefly run through how to use them with you, but everything else is pretty much used at your own risk. So just be really careful down there. Make sure you're wearing your safety glasses, all that kind of stuff. If you're sawing, if you're drilling, make sure you're using clamps appropriately so things don't fly around the room. And whenever you borrow the key from 3116, just say, like, Justin has key, and give your phone number so if somebody can't find the key later in the day, they can call you. I'm sure you guys all have each other's phone numbers by now, but just so that we can keep track of the key. So these are the due dates. You'll probably start building sometime next week, or at least gathering your supplies and planning. You've already started the decellularization, you're going to continue that, and then we'll confirm it around week 10 with a simple Daffy stain. And that's the week that you'll turn in your bioreactors. So that Friday, you'll leave them in the lab, and Haley and I will come in and just inspect them, make sure that they look good, make sure that they turn on and work. And if they do, great. If they don't, we'll tell you, and you'll have the weekend to fix it. And then week 11, I think on Wednesday, is when we actually start stretching them. So that'll be kind of the do or die moment for your bioreactor. So you'll want to make sure that you're ready then.
Is that the week after Thanksgiving?
Yeah, I think so. And so everything is kind of work at your own pace. If you want to come in on nights or weekends, that's fine too. You have access to the building and the lab. As long as you have a buddy with you, you can use the lab. All right. So switching gears a little bit into what we're going to do for the analysis of the lab. First of all, we want to know, what are ligaments made of? What are we targeting? What is an important signal for making ligament? What's kind of a hallmark of success, which is what we talk about a lot in this class? What would be a bad sign? What would be something that would happen if it wasn't going to be a ligament? And then we're going to design a primer for each of those, and we'll have a control, what we call a housekeeping primer. I'm going to turn this on. So that we can do PCR and look at our analysis for mRNA expression in the ligaments. So I'm going to run you through how to make a primer right now so you can kind of see it. You guys are going to be in charge of actually figuring out what you want to tag. What's going to be a good hallmark, what's going to be a bad hallmark, and then I'll give you some suggestions for housekeepers. We'll probably all use the same housekeeper gene just to be consistent, and that'll probably be like GAP TH, which is in every cell all the time. So it's just a good benchmark of this worked or this didn't. So I have some of these open already in a browser. Just pull this out over here. Okay, so if you go to NCBI, have you guys been to NCBI before? And you're going to type in what you want to find. So pro-collagen type 1. And hit search. It's going to come up with all sorts of results. There's lots of articles about pro-collagen type 1. There's all sorts of epigenomic data, but what we want is the nucleotide sequence record for whatever we're looking for. So in this case, pro-collagen type 1. So we're going to click on that, and it gave us 18. 18 is actually a really low number. You're usually going to get a lot, on the order of maybe 100 or so. So what we want to do is try to look for a pro-collagen type 1 in a species that's going to be helpful for us. So we have dog ligaments, so we don't want dog because there's not going to be anything expressed there. But we're going to be using 3T3s. And what are 3T3s from? Nice. So musculus is a mouse. Mus is in Latin. So we want to look for mouse sequences of pro-collagen mRNA. So if we look at this one, it says mouse, pro-collagen, endopeptide enhancer protein. That's not exactly what we want to look for, but it does come up as a homologous search. So now we have human pro-collagen type 1. Well, this is what we want, pro-collagen type 1 alpha, but this is human. And we don't know how homologous those are, or how similar they are, so we're not going to click on it yet. However, if you don't find a mouse, you can run a blast search between mouse and human and see how similar they're supposed to be. And then if you can't find a mouse one for what you're looking for, but you find a human and they're greater than 70% homologous, which is kind of a baseline, then you can use whatever species you can find. Let's see here. Pro-collagen type 1 human, mouse pro-alpha pro-collagen gene, mouse DNA sequence, human. Okay, so it's one here. This is a really long sequence. So this is actually the DNA sequence of the pro-alpha pro-collagen gene. It's pretty close to what we're looking for though. So we're going to go ahead and click on it. It's going to give us a lot of information about it. So it tells us what it is. It's the mouse pro-alpha pro-collagen gene. It tells us the keyword collagen. It tells us that it's a mouse. It's actually a house mouse. Let's see here. I can tell you all sorts of information. This is the FASTA format sequence for it. You guys familiar with that? So FASTA format doesn't use the GTCs and A's. It uses a different kind of abbreviation mechanism to tell you the amino acid sequence instead of the actual gene sequence. So you can use FASTA format, but we're going to scroll down and try to find the sequence. So here it says origin. This is the entire DNA sequence for procollagen type 1 alpha in a mouse. So we're going to copy this in all of its entirety. All the way up here. We're just going to copy that. Then we're going to go to something called primer 3, which look at this. It says pick primers from a DNA sequence, which is exactly what we want to do. So we just paste what we copied. It's okay if the numbers are there, but you can see here it also says paste as source sequence below, 5 to 3 prime. You can also use FASTA format, so if that's all you could find, you could do it. But basically this is what you want to paste there, the GTCA sequence that you find. And then you're going to say pick left primer and pick right primer. If you found a paper that already used a primer and they had really good success, you could paste those primer strands in here and make sure that it matched your sequence. So that would be one way to go about it. But we don't know anything about this sequence, so we're going to go ahead and ask it to pick some primers for us. Another thing you could do, if you had certain targets that you wanted, again, if you found a paper that said this is the key to getting a really good pro-collagen primer, then you could put that in there. But we really just don't know that much about it. So this is where you want to come down here. General primer picking conditions. Primer size between 18 and 27, that's pretty standard. Optimum is 20. I usually end up between 20 and 22 for a good primer because you need it to be long enough that it's going to be specific, but short enough that it's not going to have trouble binding, right? Because these are really fast reactions that are happening in a PCR machine. The temperatures, you don't have to go in and change these, but you do need to make sure you remember what it was because you're going to want to write down what the actual primer temperature is because when we're making our PCR sequence, it's really important that you know what it's going to melt at, right? Because we have to melt it to get it to bond to everything else. The rest of this you really don't need to worry too much about, so just be mindful of the size and the temperature. These are the defaults. You don't have to change them, but you could. So we're going to hit pick primers. It's going to run a little algorithm and try to find some 20-ish sequences that match. So here's what it comes with. Left primer, right primer. It tells you where in the sequence it starts, the length of it, the melting temperature. This is here it is here, the percent of GRC bases in the primer. So 60% is fine. Anything over 50 is probably going to be good. It tells you the 3 or the 5, right? That's good. And it gives you the actual sequence. If you scroll down, it'll actually show you where it is. So here's the left primer. Here's the right primer. Barely see what I'm doing. There you go. So in between the left and the right primer, or where the arrows are, this is that sequence that we are going to match. So in between the left and the right. So what this program is saying is that this left and right primer sequence combination should be specific enough to only bind 2 sections of mouse procollagen type 1 alpha mRNA. It's pretty cool, right? That something took us 3 steps, and we found something that is going to be that specific. And also, it'll give you the first one. This isn't necessarily like this is the best primer here at the top. It's going to give you a lot of options at the bottom as well. So these are additional oligos that also fit the same profile. So if you look at them, they're all 20 because we told it that's our optimum, so it was able to do that. It'll give us the melting temperature, so they're all pretty similar. We're getting a little bit lower in the GC percentage. And then if we look at the sequences, it looks, we can't tell a lot from the sequences. So the only reason why you might want to pick one of these over the one at the top is if you know something about your sequence. So this is a long sequence. I'm going to bet we don't know anything about it because it's really, really long. But in my PhD work, we had sequences that were maybe only like 400 to 700 base pairs, so much shorter. And in those, you wanted to stay away from the ends for sure. You wanted to be kind of in the middle to make sure you didn't get any sort of end effect. And then we also would do certain ones that were similar together. So we would do collagen type 2, collagen type 1, pro-collagen type 1, tenascin, so lots of different ECM proteins that are kind of similar. And so I would run those blast searches comparing the sequences to see how homologous they are, and then run my primers comparing all of my primers together to see how similar they were. And then sometimes I'd be forced to pick maybe the second or third one on the list there because it was less likely to accidentally probe something else. So it only tells you that this is a really good probe for this sequence, but it doesn't tell you, oh, if you're tagging something else similar, you shouldn't use it. That's really up to the designer to know. And it's something that you wouldn't know necessarily right now, but if you got this as an everyday job, as your part of your dissertation, you would know to blast everything and make sure you weren't accidentally tagging the wrong thing. So in your case, you're going to do something that you think is pro-ligament, something that's going to be helpful for making ligament, something that's not going to be helpful for making ligament. So what type of mRNAs do you think might be not helpful for making ligament?
I think there are characteristics of other adipocytes.
Yeah, adipocytes, right? A lot of cells revert back into fat when they're unhappy or stressed or something like that. So that could be something you could look for, a marker for adipocytes. Any markers for, say, calcification? That's something that happens in the body, right? Sometimes calcification happens. And if we're too strained, we might be mimicking more of a high strain. It might think, oh, I'm supposed to be a bone cell. We could develop more like scar tissue type of tissue, which is a fine line in ligament because it's collagen 1 anyway. But that's something that could come out of it. It could be too much collagen. Then, elastin, you don't want too much elastin, but you want some elastin. So you'll want to look up some papers and see what have people looked for as positive progenitors of ligament and what have people looked at as kind of antagonists to ligament. And there's lots of papers out there. I've done a few quick searches, so you should definitely be able to find some papers.
Question on this. So we are using the pro-collagen gene, a bunch of introns and exons. How do we know what we just preferenced? I mean, because we're not preferencing the mRNA right now, we're preferencing the DNA.
Right.
And we're not even sure if it's an exon.
Right.
So how can we justify this?
So you can go, oops, lost my mouse. You can go back here, and it tells you where the introns and exons are, and you can make sure that you're not within that area or that you are within that area if you want to.
Because each one of those exons should be for pro-collagen.
Right. Yeah. So again, this one's not ideal, but it's a pretty good match. So let's see if we can try it.
I mean, it has the information within it, but it's just
Right.
making sure we're referencing the right information.
I think this one's even worse. Yeah. Let's look at this one just for fun. this one is a fragment of a specific type of mRNA. So, but the primer 3 is made to get the sequence from DNA or from existing mRNA information. It can do it either way. It should be pretty accurate. The last step is to go to IDT, which is Integrated DNA Technologies, and we're actually going to order our primers from here. Yeah. So what we'll actually do is go in here and do a single entry. And then all you have to do is copy the primer that you want to make. So this one. Over. And it tells you what it's going to make. It's a standard desalting, normalization, add to order. And then it's going to make the primer for us. Please do not enter instructions or modifications. Okay. And so I'll actually order these for you once you tell me what you want. They're fairly cheap, usually less than $10 each. They come as a desalted, like lyophilization powder that you bring up in water, pretty simple, and then you add it to your PCR product when you're making your PCR product. Or you add it to your solution when you make your PCR product. So we'll order these for you once you decide on your primers. And not every primer is going to be absolutely perfect. These things sometimes take time. So if you don't get a successful primer, it's going to be okay. But it's really just doing the research on what you want to tag, going through these steps, making the primer, ordering the primer, and then doing the PCR. And then we're actually going to yeah. So afterwards, we're gonna we have a PCR in our lab. We're gonna make cDNA. We're gonna make mRNA, and then we're going to add the primers into the solution and run it through a cycle of basically annealing and then thawing and annealing and thawing. And so it's 60, 70 sequences of melting and reforming, and so it'll multiply our PCR product so that we can actually see it, get it to a number we can see on a gel. So you guys have run simple electrophoresis gels in 202 lab. This is going to be very similar to that. We're going to have a gel, we're going to input a ladder. The ladder will help us separate out the base pairs so that we can figure out what we're looking at. So you do need to know how many base pairs your mRNA is supposed to be. I'm guessing most of these are going to be around 200-ish, 300-ish, because they're going to be kind of small, like procollagen, collagen, tenacin, those are all around that range. So you'll have a few wells that you're going to fill in. So you'll have, of course, the ladder, you'll have your pro ligament, your anti ligament, and then a housekeeper. And so you'll run those 4 lanes and then take a picture of your gel like this. And we're going to use a UV reactive stain again, but we're also going to physically stain it with something called fast stain blue so that we can see it right as soon as it's done running and see if we had a successful run. Because if not, you usually have enough PCR product to run another one and you can run it really quick. So this is an example of what you'll put in your report at the end. So you'll have some pictures of the ligament, certainly some pictures of it in the process, but not really immunohistochemistry this time. We're going to do PCR. And there are also, if you have your notebook with you, there's some pictures of the PCR cycle that it'll go through. That would be a good thing to know how to explain in your report so you kind of know how to say, well, I chose this melting temperature because then we programmed a 62 Celsius melt in our PCR cycle, just so you kind of understand why we would pick that cycle. So you guys have questions about the analysis we're going to do for this one? It's really different than what we've done before.
So we're going to take the cells and then create cDNA and then create mRNA, or we're going to PCR the cDNA?
We're going to PCR the cDNA to make the mRNA.
So that's why we're okay creating primers
For the
bind
CD.
to the
Yeah.
DNA of the thousand.
Yeah. It's a good question.
Switch all the T's to U's.
Yeah, that's a good question. Yeah, we're gonna do it by hand. No, I'm just kidding. We're gonna do it in the PCR machine. It'll do it all for us. That's why we're binding the DNA product. So,
So PCR will do the cDNA as well. The PCR machine, or we
the PCR machine does the cDNA. It's just a separate reaction. Yeah, it's pretty simple. For each of those two processes, there's a little kit that has 4 vials in it, and it's add 1 microliter of this, 10 of this, and 5 of this, and put it in the tube, and put it in the PCR, and you run it overnight. It's pretty easy. It's just a step you have to do. Yeah. So we'll make our cDNA. In order to do that, we of course have to grind up our ligaments. So that's always fun. So we homogenize it, pulse it up with a little blender. Pretty fun. And then get all the cells out of it and run it through cDNA. So if you haven't done it before, it's not that hard, but it's a pretty common lab procedure that you'll end up doing eventually. So I think it's really realistic for grad school. I did it at least weekly, if not more. So it's pretty useful. Okay, so next week we are going to review for the midterm. It's going to be a pretty open format Q&A. You guys are going to lead the discussion, ask questions about whatever you have questions about still. I'll put up some topics, but otherwise just Q&A about the midterm. And the midterm will be in class 11:05 at 9, so it'll be in here. And it'll cover material from your lab notebook, things that you've read about, experiences in lab, but really the focus is on technique and rationale. Why are we doing what we're doing? How are we doing what we're doing? Because those are important kind of senior level skills you should have, opposed to the type of skill set you have when you were in 202 and just kind of going through the motions. You guys should really understand the how and why are we doing this. So if you have any questions before then, let me know, but definitely bring your questions to class next time because it's going to be a pretty open discussion. You guys can just kind of drill me about whatever you want to know before the midterm. And then of course, next week we have our AFM practicals scheduled. So if you don't remember when yours is, let me know, but we schedule them all. And you'll have up to an hour, but you're just going to have to get in contact with the surface, do a scan, and prove that you understand the basics of AFM. Okay? All right. If you don't have any questions, feel free to come up again and look at the bioreactor. And if you want to go next door and check your cells,